When you reward yourself with a sweet treat, chances are you've just consumed a lot of this 6-carbon sugar. Fructose, also known as fruit sugar, derives its name from the Latin word fructus, meaning fruit. It is the only naturally occurring ketohexose, and is commonly found in honey, as well as a variety of fruits and vegetables. Our bodies metabolize fructose by modifying it so it can enter glycolysis, or through a separate pathway for fructose metabolism. These processes are mediated by a series of metabolic enzymes, but today we'll be focusing on aldolase. So what exactly is aldolase? Fructose 1,6-bisphosphate aldolase, referred to sometimes as simply aldolase, catalyzes the cleavage of fructose 1,6-bisphosphate into two 3-carbon triose phosphates, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. The reaction is driven forward by mass action, so it is important that the concentration of G3P in the cell remains low. The enzyme also readily catalyzes a reverse reaction, an aldol condensation, which is where it gets its name, aldolase. Aldolase plays a central role in not only glycolysis, but also in gluconeogenesis, as well as fructose metabolism. There are 3 different classes of aldolases: Class 1, Class 1A, and Class 2. Class 1 aldolases typically form homotetramers, with a total molecular weight of approximately 160 kilodaltons. These aldolases utilize the lysine residue within the active site to create a stabilizing Schiff-based intermediate with a substrate. The enzyme shown here is found in our muscle cells and captures the lysine residue 229 covalently bonded to the sugar, when the sugar molecule has been cleaved, leaving only half in the active site. Class 2 aldolases, on the other hand, are metal-dependent and rely on divalent ions, typically zinc 2+, to stabilize the carbanion intermediate formed by the substrate within the active site. Class 2 aldolases typically form homodimers with a total molecular weight of approximately 78 kilodaltons, but can also form tetramers and octamers. Class 1 aldolases are typically found in higher eukaryotic organisms, like plants and animals, while Class 2 aldolases are more common in morphologically less complex eukaryotic organisms, such as protozoans, fungi, yeast, and algae, in prokaryotes as well as some archaea. Finally, Class 1A aldolases are found in archaea, and have a similar mechanism of action as Class 1 aldolases. They have been reported to exist as homooctamers, decamers, or even higher oligomers. Class 1 aldolases in vertebrates exist as 3 isozymes, with different tissue distributions and kinetics. Aldolase A in muscles and erythrocytes, aldolase B in liver, kidney, and small intestine, and aldolase C in brain and neuronal tissue. Deficiencies in aldolase lead to certain genetic disorders, such as hereditary fructose intolerance, HFI, an inherited inability to digest fructose due to a deficiency in activity of aldolase B. Unlike glucose, which is metabolized directly throughout the body, fructose is predominantly metabolized in the liver. This is done through a process called fructolysis, which is directed towards replenishing liver glycogen and synthesizing triacylglycerol. In the liver, fructose is rapidly converted to fructose 1-phosphate via fructokinase. Fructose 1-phosphate is then converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by aldolase B. Individuals with HFI cannot further metabolize fructose 1-phosphate, leading to an accumulation of the compound which is toxic to the liver. Phosphate is utilized in the production of fructose 1-phosphate by fructokinase, and as fructose 1-phosphate accumulates, intracellular phosphate stores are depleted. The combined effect of this change inhibits phosphorylase A, which shuts down glycogenolysis. Glycogenolysis is the catabolism of glycogen branches due to the sequential removal of glucose monomers as glucose 1-phosphate by phosphorylasis, which is how the liver raises blood glucose levels. Aldolase B deficiency also causes impaired gluconeogenesis, since dihydroxyacetone phosphate and glyceraldehyde 3-phosphate cannot be condensed to form fructose 1,6-bisphosphate, which worsens the issue. People who have HFI usually develop a strong dislike for sweets and fruit. After eating foods containing fructose, they may experience symptoms such as severe abdominal pain, vomiting, and low blood sugar, or hypoglycemia. Aldolase also has many biotechnological applications, and serves as a potent biocatalyst in the food, pharmaceutical, and biofuel industries. Aldolase is extensively used in the food industry, particularly for the production of high fructose corn syrup. High fructose corn syrup is a widely used sweetener in food and drinks, known for its cost effectiveness and improved solubility compared to conventional sugars. In the pharmaceutical industry, the enzyme is used for the synthesis of lactulose, a widely used prebiotic synthesized through the reaction between lactose and fructose. The biofuel industry takes advantage of aldolase's ability to convert fructose 1,6-bisphosphate to DHAP and G3P, which are crucial intermediates in the production of biofuels, such as ethanol and butanol, in lysocellulosic biomass, or plant-based material. In recent years, aldolase is being looked at as a therapeutic target as researchers have found many moonlighting or non-enzymatic functions of the classical enzyme, which involve its impact on cellular scaffolding, signaling, transcription, and motility. The enzyme is rapidly developing as both a drug target and vaccine candidate, and more research is still being done in these areas. Looking back, aldolase has come a long way from being merely a protein used to catalyze a forgettable step in glycolysis of gluconeogenesis. So who's to say that aldolase isn't an interesting protein after all?